Hello all, welcome to my channel. In this video we are going to discuss about DNA structure in details and details about the double helix, functions of DNA and chargaff rule. So in the last video of genetics we are discussed about the search for genetic experiment by search for genetic material by Griffith experiment, and the conclusion is nothing but the DNA. So let's discuss what is the DNA exactly. DNA is found in the nucleus of cell, and DNA contains chromatin material with protein which carries a genetic material. The protein is nothing but the histones. DNA is made up of subunits which scientists called as nucleotides. Each nucleotide is made up of sugar, phosphate and bases. The basis is nothing but the nitrogenous bases. There are four different bases in a DNA molecule, that is adenine, cytosine, guanine and thymine, out of which adenine and guanine are the purine base and cytosine and thymine are the pyrimidine bases. The number of purine bases equal to the number of pyrimidine bases. The basic structure of a DNA molecule is helical, with the bases being stacked on the top of each other. Next is the component of DNA. As I say, DNA is a giant molecule and made up of components like nitrogenous bases, pentose sugar and phosphate group. So DNA is a polymer. The monomeric unit of DNA are nucleotides and the polymeric units are known as polynucleotides. Each nucleotide consists of 5 carbon sugar. In case of DNA, the sugar is deoxyribose sugar. A nitrogen containing bases attached to the sugar and phosphate group, and there are 4 different nucleotides. Then what are the purine and pyrimidine bases? Adenine and guanine are purines. Purines are the larger of two types of bases found in DNA. So here is the structure of a purine and pyrimidine. Here is the purine bases. These are the larger molecules. The 9 atoms that make up and fuse to form a ring. Number of 5 is the carbon and 4 is the nitrogen. Look at here, there is the 4 nitrogen and 5 carbon. Sorry for inconvenience. Look at the diagrams of purine and pyrimidine. In purine, it is larger than pyrimidine as you compare in the photographic image. And in purine, there are 9 atoms, out of which 5 is the carbon. Look at here, 1, 2, 3, 4 and 5. These are the 5 carbon and 4 nitrogen. Here is the position number 3, 1 nitrogen. Position number 1, there is another nitrogen. Position number 7, there is another nitrogen. And position number 9, there is another nitrogen. So purine is contain 4 nitrogen and 5 carbon. Total 9 number. All rings atoms lies in the same plane. Then what about pyrimidine bases? Cytosine and thymines are the pyrimidine bases. The 6 atoms are there, out of which 4 carbon, look at here, position number 4, position number 5, 6 and here in position number 2. There is 4 carbon atom and 2 nitrogen in the position number 3 and the position number 1. 2 nitrogen. So as it is 6 atoms in the ring, benzene ring, it is smaller in nature, we discussed already. So 4 carbon and 2 nitrogen in the pyrimidine basis. All pyrimidine ring atoms lie in a same plane as purine. So here we finish with purine and pyrimidine or the nitrogenous basis, the structure, how the carbon and nitrogen are attached within. Next is pentose sugar, which is nothing but the deoxyribose sugar. The deoxyribose sugar of the DNA backbone has 5 carbon and 3 oxygen. As you see in the photographic image, the carbon atom are numbered as 1, 2, 3 and 4 and 5. Distinguishing from numbering of atoms, purine and pyrimidine rings, the hydroxyl group on the 5' and 3' carbon linked to the phosphate group. Deoxyribose lack an hydroxyl group at the second position when compared to ribose. The basic difference is ribose sugar or deoxyribose sugar, or you may say DNA RNA, is the lack of one hydroxyl group within them. Next is nucleoside and nucleotides. Nucleotide consists of nucleoside and phosphoric bases or phosphoric group. Here is the nucleotide and it contains nitrogenous base, pentose sugar and phosphoric group, and nucleoside is contain nitrogenous base with pentose sugar. Next is the double helical structure of DNA. Johnson and Friedrich Mescher recognized the DNA, but the double helical structure was proposed by Watson and Crick in 1869. The double helix of DNA has 3 features, that is, first, it contains 2 polynucleotide strands wound around each other. The backbone of each consists of alternating deoxyribose and phosphate group. The phosphate group bond of the 5 carbon atom 1 and deoxyribose covalently bond to the 3 carbon atom of the next. 2 strands are parallel, as you see here, 2 strands are parallel with each other. One is 5 to 3 and another is 3 to 5 dash. The DNA strands are assembled into 5 to 3 dash direction, and by convention we read them the same way. The purine or pyrimidine attached to each other by deoxyribose project in towards the axis of the helix. Each base form hydrogen bond with the one directly opposite it, forming the base pair. That is, one purine and one another pyrimidine is there. 3.4 angstrom separate the plane in which adjacent base pair are located. 3.4 angstrom. Here is the 3.4 angstrom. The double helix make a complete turn over 10 nucleotide pairs. So each turn take a little. That is, each turn contain 3.4 angstrom. This is 3.4 angstrom. There is an average of 25 hydrogen bond within each complete turn. Each complete turn contains 25 hydrogen bond, which make them stable. The diameter of a helix is 20 angstrom or 2 nanometer. The helix can be virtually any length. When fully stretched, some DNA molecules are as much as 5 centimeters in long. Next is the function of DNA. DNA carries the genetic material. We all knows about that. Each gene serves as a recipe on how to build a protein molecule. Proteins perform important tasks for the cell function or serve as building blocks. Proteins are the building blocks we all know about. I think the flow of information from the genes determine the protein composition and thereby functions in the cell. DNA is situated in the nucleus and organized into chromosomes also. Every cell must contain the genetic information and DNA is therefore duplicated before the cell divide. It also transcribed into RNA. The RNA is first processed so that non-coding parts are removed and then transported out of the nucleus. Outside the nucleus, the proteins are built based upon the code in the mRNA. Last is chargaff rule. Edwin Chargaff, a biochemist, discovered that the number of nitrogenous bases in the DNA was present in the equal quantity. The rule states that any sample of DNA shows 1:1 ratio of pyrimidine and purine bases, that is A equal to T, G equal to C. DNA of any cell from any organism should have a 1:1 ratio of purine and pyrimidine bases. This is the rule of Edwin Chargaff. Here we finish with the structure, component of DNA and also the Chargaff rule. If you have any query, please comment in the comment box. Thank you.